Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT BET JRF Entrance Exam Question Paper Part 1. I have divided into two parts. This is the first part. Coming to the first question. A 300 base pair long B form of plasmid DNA has 20 complete turns. This DNA molecule is. So normally there are 10.5 base pair per turn. So 300 base pair DNA should have had 28.5 turns. That is 300 divided by 10.5. You will get 28.5 turns for 300 base pair DNA. That is if it was not supercoiled. So it would be 28.5 turns. But we can see that the turns have been reduced from 28.5 to 20. So it has become negatively supercoiled. So the answer here is option 2, negatively supercoiled. Next one. Which of the following sequences is a palindrome? So a palindromic sequence is a nucleic acid sequence in a double-stranded DNA whereby reading in certain direction, that is 5' to 3' direction on one strand, is identical to the sequence in the same direction in the complementary strand. That is, if you read the first sequence from in the first strand, sequence in the first strand from 5', 3' direction, the sequence in the second strand, if read in 5', 3' direction, would be the same. So here if you see, you have got ACGGATTCGC. Now if you read it in this direction, that is from 5' to 3' direction for the second strand or the complementary strand, you will see that it is GCGAATC, which is not at all identical to the first strand sequence. Similarly, in case of option B also, as well as option C also, but option 4 if you see, you can see that AGGCCT. So it reads the same in 5' direction on the first strand as well as in the second strand. So the answer is option 4. Then the nucleotide sequence in an mRNA. They have given the nucleotide sequence. Assuming that the ribosome could translate this mRNA, how many amino acids long would you expect the resulting polypeptide chain to be? So this is the sequence UAA. The beginning itself is a stop codon. You have to start from AUG. AUG up till UAG. UAG again is a stop codon here. So how many amino acids are there? 1, 2, 3, 4, 5 and 6. So how many amino acids long would the resulting polypeptide chain be is 6. Answer is option 1. Then 1 ml of NaDH solution gave an absorbance of 0.31 at 340 nanometer wavelength with 1 centimeter cuvette path length. Calculate the molarity of NaDH in this solution. So epsilon 340 is 6220 and molecular weight of NaDH is 663 Daltons. So you know the formula, A is equal to epsilon Cl. Substitute it, you will get the concentration, that is 4.98 into 10 raised to minus 5 molar. Or else you can write it as 5 into 10 raised to minus 5 molar. And 1 molar is 10 raised to 6 micromolar. So 5 into 10 raised to minus 5 molar will be 50 micromolar. So the answer is option 1. Then number of molecules present in 1 ml of 250 microgram per ml solution of a 10 kDa protein will be. They have given Avogadro number also. So 250 microgram are present in 1 ml of 10 kDa protein. So number of moles is weight by molecular weight. So 250 into 10 raised to minus 6 grams divided by 1000 Daltons. So you will get a value over there. And 1 mole has Avogadro number molecules per mole. So 250 into 10 raised to minus 10 moles has 250 into 10 raised to minus 10 into Avogadro number. That will give you 1.505 into 10 raised to 16. So that is option 1. Then next one. The two most common processes that lead to production of multiple functional proteins from the same DNA sequences are. So the answer is RNA editing and alternative splicing. Now what is RNA editing? So RNA editing, it is a molecular process in which the information content is altered in the RNA molecule. Such changes have been observed in tRNA, rRNA, mRNA of eukaryotes but not in case of prokaryotes. And this editing can be either deletion or an insertion. Then alternative splicing is the process of selecting different combination of splice sites within an mRNA precursor to produce variably spliced mRNA. These multiple mRNAs can encode proteins that vary in their sequence and activity and yet arise from a single gene. Even RNA editing also, once you insert a new nucleotide or delete a nucleotide during RNA editing, you get a different RNA sequence but produced from the same gene. So it will give rise to different proteins. So RNA editing and alternative splicing are the answer. The next one. Which of the following statement is false? The potassium ion channel allows potassium ion through the plasma membrane. Prions allow chloride ion to pass through the plasma membrane. That is not correct. Prions allow the passage of small macromolecules such as glucose or maltose from the external environment to the cell interior. So the answer which is false is option 2. That is correct also for as per the question. Then next one. Which of the following statements about the glycolysis pathway in the cytosol is incorrect? It makes ATP. Yes, it makes acetyl-CoA. It doesn't make acetyl-CoA. Glycolysis pathway doesn't make acetyl-CoA. So answer is option 2. Then the tryptophan operon is transcribed when tryptophan concentration is high in the cell. Then no need of any tryptophan biosynthesis. The tryptophan repressor is bound to tryptophan or a similar shaped molecule. That time it will bind to the operator and mRNA will not be synthesized. So it is not transcription of tryptophan biosynthetic genes. Tryptophan bound to its aporepressor. So tryptophan, if it is bound to its aporepressor, it is the same thing as option 2. It will go and bind to the operator region. Then the appropriate co-repressor is absent. So when tryptophan is absent, that is co-repressor, that is tryptophan. So tryptophan is absent. It will result in the synthesis of the proteins required for tryptophan biosynthesis. So answer is option 4. Then glycosylation of a protein occurs in endoplasmic reticulum. I repeated the same question. It is an typographical error. Okay. Next one. Innate immunity is mediated by. So innate immunity, it is mediated by toll-like receptors. Then humans have 23 pair of chromosomes, while our closest relatives, that is chimpanzees, have 24. Chromosome studies indicate that at some point early in human evolution, the two chromosomes simultaneously broke into a larger portion and a small portion. The large parts combined to form large chromosome and a small parts combined to form smaller chromosome, which was subsequently lost. The important chromosomal change can be best described as, it is nothing but translocation. So translocation, it is a type of chromosomal abnormality in which a chromosome breaks and a portion of it reattaches to a different chromosome. So here it is translocation followed by deletion. There is translocation, then later the smaller parts combine to form smaller chromosome, which is subsequently lost. So there is deletion also. So answer is option 2. Then transgenic organism carry the trans gene in. So it carries in both gametes and somatic cells. What is a pseudogene? A coding region that cannot be translated into a functional protein. It is called a pseudogene. An operon is promoter, an operator, and a group of linked structural genes. Answer is option 4. Pyrosequencing, that is a type of DNA sequencing, uses which of the following? It uses emulsion PCR. Answer is option 1. Then necrotrophic plant pathogens. So they are pathogens which kill cells of the host plants. Answer is option 1. Transgenic crops occupying the largest cultivated area in the world are tolerant to. So they are tolerant to herbicide. I think having largest cultivated area is soybean, I think. Transgenic crop. Okay. You can search the internet for that. Then next one. Which one of the following phytochrome gene in Arabidopsis thaliana is responsible for hypocotyl elongation, flowering and seed germination? So it is PHYB. Then bread wheat triticum aestivum is an allohexaploid. The cytokinin receptor is cytokinin is a plant hormone. The receptor is a 2-component histidine kinase. So it is a 2-component histidine kinase. Then the BR2 brachytic 2 gene encodes a P-glycoprotein required for normal auxin transport in cone. Which one of the following describes the phenotype of BR2 mutants? So BR2 mutant plants, they are characterized by shortening of lower stalk internodes, unusual stalk strength and tolerance to wind lodging, darker leaves persisting longer in the active green state in comparison to wild type plants. So they are having short internodes. So out of the options here, short internodes is correct. Answer is option 2. Then what is the generation time of a bacterial population that increases from 100 cell to 100,000 cells in 3 hours of growth? So n is equal to n0 2 raised to n, where n0 is the initial population, n is the final population, small n is the number of generation. So from the, if you know n and n0 and you can calculate for n. So n is equal to 10. Now substitute in the formula t is equal to t into n, where t, capital T is total time and small t is generation time and small n is number of generation. So that is, capital T is 180 minutes. 3 hours is 180 minutes. That is equal to t into 10. So option 2, that is 18 minutes is correct. Then, which of the following is the most populous in the CNS? So it is granule cells. Answer is option 3. Huntington's disease is caused by degeneration of neurons in the dash, leading clinically to involuntary movements, chorea, psychiatric symptoms and dementia. So it is basal ganglia followed by cortical regions. The next one. The function of pyloric sphincter is to prevent the backflow of material from duodenum to the stomach. Answer is option 2. Which of the following types of stem cell have the highest risk of teratoma formation? So it is embryonic stem cells. Then, which one of the following cell type is the most characteristic component of the early stage of acute inflammatory reaction? So it is neutrophil. The first cell attached to site of injury are neutrophils. Then it is followed by monocytes, then natural killer cells, T cells, B cells, mast cells, etc. So the first one to occupy will be neutrophils. Answer is option 2. Then severe combined immunodeficiency mice and nude mice differ in which of the following cellular components? So SCID mice have genetic immune deficiency that affects their B and T cell. Then nude mice have deteriorated or absent thymus, resulting in reduced number of T cells. So nude mice have B lymphocytes and skid mice do not have. So nude mice have B lymphocytes. So answer is option 1, that is B lymphocytes. Then smallest lipid containing enveloped animal virus belongs to Flaviviridae. Answer is option 3. Which of the following is the characteristic cell found in granulomatous inflammation? So granulomatous inflammation is characterized by the formation of distinct granulomas composed of aggregates of epithelioid histocytes with a peripheral cuff of lymphocytes and plasma cells and occasionally a necrotic center. So which of the following is characteristic cell found in granulomatous infection is plasma cell. Answer is option 2. Role of diatoms in ocean is primary production. Then one of the following is free living aerobic nitrogen fixing bacterium in the oceans. It is Azotobacter. Then next one. The prominent group of microorganisms involved in marine biocorrosion is. So it is sulfate reducing bacteria. Greenhouse effect with respect to global warming refers to warming effect. Answer is option 2. A high BOD value in aquatic environment is indicative of. It is indicative of highly polluted system due to excess of nutrients. Primary productivity at the climax stage of a succession is. It is equal to consumption. Then you have isolated an Indian strain of phage CSIR. You measure the nucleotide base content of the phage and find the following results. A is 40%, G is 10%. What are the likely percentage content of T and C? Now phage CSIR, it is having single-stranded DNA. So you cannot calculate how much T and C will be present from the given data because single-stranded DNA doesn't follow the Chargaff rule. So answer is it cannot be predicted from the given data. Then cluster analysis in DNA microarray experiments refer to. So a DNA microarray, it is a collection of microscopic DNA spots attached to a solid surface. Scientists use DNA microarrays to measure the expression level of large number of genes simultaneously or to genotype multiple regions of a genome. So cluster analysis in DNA microarray is genes that are likely to work in concert in the cell. Answer is option 3. Then next one. A sample of a homomultimeric protein containing one atom of iron per polypeptide, which amounts to 0.56% by weight. Gel filtration indicates that the molecular weight of the multimer is 20 kDa. The maximum number of subunits that the protein may have, assuming that the atomic weight of iron is 56. So 0.56% by weight means 0.56 by 100 into 20,000. So atomic weight of iron they have given as 56. So 112 divided by 56 is equal to 2. Answer is option 3. Then next one. A tripeptide has an amino acid composition lysine, phenylalanine, proline. Dancyl chloride treatment produced DNS-PHE. The peptide is not cleaved by trypsin. The primary structure of the peptide is. So dancyl chloride reacts with the free amino groups of peptides and proteins. That is N-terminal proteins. N-terminal region. Okay. So phenylalanine will be the first one. Then it is not cleaved by trypsin. So trypsin cleaves a peptide bond between carboxyl group of arginine or the carboxyl group of lysine and the amino group of the adjacent amino acid but not before proline. So lysine have to come after proline. So here, as per the question, phenylalanine is the N-terminal amino acid. The question says that it is not cleaved by trypsin, so lysine has to be at the C-terminal end, and in between it will be proline. So it will not cleave the lysine because proline comes before lysine. So the answer will be phenylalanine, proline and lysine. Then next one. You have been given two unlabeled samples of PTH lysine which has been derivatized either in the alpha NH2 or in the epsilon NH2 group. Which of the following techniques will be used to distinguish between the two? So alpha NH2 will have a pKa value of 8.95 and the epsilon NH2 group it will have a pKa of around 10. So the option that can be used to distinguish between the two is pH titrations. Answer is option 1. Then polytene chromosomes is generated due to. So polytene chromosome is generated due to repeated DNA replication in DNA without segregation. Then, if a man of blood group AB marries a woman of blood group A whose father was of blood group O, to what different blood groups can this man and woman expect their children to belong? Now, this was a regular question not only for DBT exam but also for other PhD exams I have observed. This question you can refer to my channel for various question papers. I have seen this question being asked. So genotype of the woman whose father is blood group O will be AO. Now a cross between AB and AO you will get three blood groups, that is A, AB and B. Answer is option 1. A human male carrying an allele for a trait on the X chromosome is. So he is hemizygous. Answer is option 1. Positive feedback is operating. So positive feedback occurs to increase the change or increase the output. The result of a reaction is amplified to make it occur quickly. So coming to the first one. When adenosine monophosphate activate phosphofructokinase. Phosphofructokinase activity in all organisms is controlled by negative feedback loop created by the relative abundance of ATP to its hydrolysis products ADP and AMP. So their negative feedback loop is operating. When CAMP activates transcription of the lac operon, not CAMP but absence of glucose. As per me, that is the correct for this particular option. Then when tryptophan inhibits transcription of the TRP operon, tryptophan is a negative feedback. Then when N-acyl HSL promote transcription of the Lux operon. So that is the one where positive feedback is operating. So answer is option 4. Then consider a typical hepatocyte. The major cell in the liver. It is roughly a cube of 15 micrometer on a side. Assume the density of the cell as 1.03 grams per ml and 20% of total weight, which is occupied by a protein which is having 400 amino acids. The total number of molecules of that protein present in the hepatocyte will be. So cube of 15 centimeter cube on a side. So volume of the cell will be a cube. So 15 into 15 into 15 into 10 raised to minus 4 a whole raised to 3. So that will give you 3,375 into 10 raised to minus 12 centimeter cube. Now 1 micrometer is 10 raised to minus 4 centimeter. So 3,375 into 10 raised to minus 12 centimeter cube. 1 ml is 1 centimeter cube, that is 3,375 into 10 raised to minus 12 ml. Now they have given density of the cell as 1.03 grams per ml. Now 1 ml weighs 1.03 grams. So 3,375 into 10 raised to minus 12 will weigh into 1.03. So that will give you 3.47625 into 10 raised to minus 9 gram. Now 20% of the total weight of which is occupied by proteins. So 20% of the weight, that is 3.47 into 10 raised to minus 9 into 20 by 100. So one of the protein occupy mass by molecular mass. So 3.47 into 10 raised to minus 9 into 20 divided by 100 into 50,000. So 1 mole has Avogadro number of molecules. So substitution followed by calculation, you will get 8.3 into 10 raised to 9. Answer is option 1. Then next one. Process of formation of ATP from ADP while harvesting the photon is referred to as, photophosphorylation. Answer is option 1. Then match the enzyme from group A with the respective class in group B. So cytochrome P450, oxidoreductase, alkaline phosphatase, hydrolase, phosphoglucomutase, isomerase, hexokinase, transferase. So answer is option 3, 1B, 2A, 3D, 4C. Then match the cofactor in group A with the appropriate group carried in activated form in group B. So FMNH2, it is electron, lipoamide acid, thymine pyrophosphate, aldehyde, S-adenosyl methionine, methyl. So answer is option 1, 1D, 2A, 3C, 4B. There are 4 conserved homologous motifs within the BCl2 family, BH1, BH2, BH3, BH4. Which among these is critical for BCl2 family heterodimerization? So it is BH3. What is the charge of the protein having pH less than its Pi value? So when pH is less than Pi, there is an excess amount of H positive ions in the solution. The excess H positive is attracted to the negatively charged carboxylate ion, resulting in its protonation. The carboxylate ion is protonated, making it neutral, leaving only a positive charge on the amide group. So pH less than Pi means the protein will be having positive charge. Then ammonium sulfate is the most suitable salt for protein precipitation because it is cosmotrophic and falls on the left side of Hofmeister series. Now cosmotrophic causes water molecule to favorably interact, which in effect stabilizes intramolecular interactions in the macromolecules such as proteins. Then ammonium sulfate is the traditional cosmotrophic salt for salting out of protein from an aqueous solution. And the Hofmeister series is a classification of ions in order of their ability to salt out or salt in proteins. Next one. There are 2 protein molecules S and M. They have the same molecular weight, same charge and structurally very similar but vary in certain domains. How can you separate S and M? So they have the same molecular weight, not polyacrylamide gel, same charge, not ion exchange, and structurally very similar but to vary in certain domains. So naturally affinity chromatography. The proteins may have difference in their affinity. So answer will be option 1. Then next one. What provides the information necessary to specify the 3-dimensional shape of a protein? So the protein's amino acid sequence, that is the primary sequence of the protein, will determine the 3-dimensional shape of the protein. So answer is option 3. Those portions of a transmembrane protein that crosses the lipid bilayer usually consist of which secondary structures? So it has a beta sheet with the mostly polar side chains, a beta sheet with mostly non-polar side chains, a helix with mostly polar side chains and helix with mostly non-polar side chains. So those portions of the transmembrane that crosses the lipid bilayer. They usually have helix and they are present in the lipid bilayer, so they will have non-polar side chains. So answer will be option 4. So coming to the next question, which one among the following suits to protein families? So it is what evolutionary related proteins that are similar in amino acid sequence and three-dimensional conformation. Option 3. Which of the following determine the specificity of an antibody towards an antigen? The amino acid loop in its variable domain. Then allosteric inhibitor affects the active site of an enzyme by which one of the following? It binds to a second site resulting in conformational change which makes the active site of the enzyme less accommodating to the substrate. Then phosphorylation controls the protein activity by which of the following reasons? So it adds energy to protein. It can induce conformational changes in target protein. Yes, that is correct. Then next one. Which of the following classes of enzymes add a phosphate group to another protein? So it is transferase. The reaction between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate to form fructose 1,6-biphosphate is best described as an aldol condensation reaction. Answer is option 1. So coming to the next question, in the following options, which one is the most reduced form of carbon? It is RCH3. The similarities between mitogenic stimulation by EGF and depolarization of the membrane of skeletal muscle cell by acetylcholine are. They are ligand-mediated conformational change in the receptor of responding cell. Then KDL sequence of the endoplasmic reticulum luminal protein is responsible for. It is responsible for retrieval of endoplasmic reticulum luminal protein from the Golgi. The direction of chemical reaction is best predicted by. It is predicted by free energy change. Then 1 gram of rice flour from 1 kilogram pack is mixed in 100 ml of sterile water. Aliquot from this solution was dispensed in test tube in order to make dilutions of 5 times or 5x to make final volume of 10 ml. 1 ml of this solution was then poured on sterile nutrient agar plate. 5 colonies were observed after 24 hours of incubation. What is the viable count of bacterial cell in the rice flour sample? So 1 gram of rice flour from 1 kilogram pack was taken, mixed in 100 ml water. The 100 ml was made up to 500 ml and 10 ml was taken from this 500 ml. So the dilution factor is 50 and 5 colonies were obtained. So 5 into 50 into 10 raised to 3 into 100 will give you 25 followed by 5 zeros. So the answer here will be option 2. Then which of the following is false about E. coli lacapron? It is polycistronic. It is an example of negative control. The presence of lactose acts as an inducer. All 3 are correct. The repressor binds to the promoter. The repressor binds to the operator, not to the promoter. So option 4 is not false. Then most of the dry mass in a tree trunk is originally derived from. So the mass of the tree is primarily carbon. The carbon comes from carbon dioxide used during photosynthesis. So answer is option 2. Then next one. You have examined unit 10,000 cells in a culture and found that only 2 cells were in mitosis. Therefore, the mitotic index is. So mitotic index is the number of cells undergoing mitosis divided by the total number of cells. So 2 divided by 10,000 you will get 0.0002. Answer is option 2. Next one. In vitro coupled transcription and translation system have been developed that use reticulocyte or wheat germ lysates to specifically translate a defined cDNA. In this system, in this technique, you have translated a gene of your interest. However, when you run SDS page to check for the translated product, you see many bands from top to the bottom of the gel. This could be due to the following reason. The translated protein got degraded. Since it is an in vitro system, it could read the cDNA in all the frames. There are a number of endogenous proteins present in the wheat germ and reticulocyte lysate. Yes, that is more correct. Option 3. Cell cycle controller is robust and adaptable. Additionally, it functions like a switch to ensure unidirectional cell cycle. This switch-like behavior can be achieved by which one of the following mechanisms? So it is for positive feedback loops to regulate the activity of cyclin-dependent kinases. Then colchicine treatment blocks the polymerization of microtubules, whereas Taxol block the depolymerization of the microtubules. Treatment with either colchicine or Taxol ultimately results in cell death. It is possible to achieve the same result even though the actions of both the agents are opposite because block transcription of microtubule because they block the transcription of microtubules. Answer is option 2. The next one. In mammals, average length of transcription factor binding sequences are quite short, that is approximately 7 base pair. Hence, the frequency of occurrence of such sites in the entire genome is very high, that is vis-a-vis the total number of genes present therein. However, the specificity of transcription is still achieved because of pairwise occurrence of those target sites in the promoters. Answer is option 2. Then next one. Cell division cycle is divided into 4 phases, G1S, G2 and M. Standard eukaryotic cell cycle are 12 hours or longer duration. Early embryonic cell cycles are extremely rapid, having time duration of less than an hour. Which of the following phases are drastically reduced in embryonic cell cycle? That is G1 and G2 phases are reduced in embryonic cell cycles. Answer is option 1. Then recognition of intracellular pathogens in innate immune cells involves node-like receptors, NLRs. Then in complementation test, Benzer simultaneously infected E. coli cells with 2 phages, each of which carried different mutation. What conclusion did he make when the progeny phage produced normal plaques? The mutation occurred at different loci. That was the conclusion that he made when the progeny phage produced normal plaques. Then discontinuous replication is the result of which property of DNA? So it is antiparallel nucleotide strands. Then primers are synthesized where on the lagging strand. So primers at the beginning of every Okazaki fragment. Answer is option 3. Which one of the following is the difference between the core promoter and the regulatory promoter? So only the core promoter has consensus sequence. The regulatory promoter is further upstream of the gene. Transcription factors bind to the core promoter. Transcriptional activator promotes bind to the regulatory promoter. So both 2 and 3 are correct. Then in tryptophan operon, what happens to the tryptophan operon in the absence of tryptophan? So in the absence of tryptophan, it cannot bind to the operator and transcription take place. Tryptophan is a co-repressor, so it cannot bind with the repressor and it will not be able to attach to the operator. So transcription of tryptophan biosynthesis gene take place. Which of the following is the correct order of mutagenesis screen? So mutagenesis, identify mutants, verify genetic bases and then positional cloning. Answer is option 3. Then next one. In area of high GC content of the human genome, gene density tend to be high. Answer is option 2. Then the bases A, G, U, C, I, INOC all occur at 5' position of the anticodons in tRNAs. What is the minimum number of tRNAs required to recognize all codons of amino acids specified by codons with complete degeneracy? That is 2. Answer is option 2. Which of the following cell surface markers is used to identify the B cells from blood samples? So it is CD19. Then administration of the DBT vaccine, diphtheria, pertussis, tetanus, would stimulate which of the following types of immunity? So it is artificial and active immunity will be stimulated. Loss of which of the following classes of molecules on the surface of a tumor cell target would result in reduced susceptibility to killing by host immune cell? So it is MHC class 1. Genes for 16S and 28S rRNA are transcribed by. They are transcribed by RNA polymerase 1. Then next one. In cancer condition, genes can be either repressed or overexpressed. Repression of genes by DNA methylation depends on high CpG density and promoter strength. Which type of inhibition requires binding of one or more substrates to enzyme before the inhibitor can bind? So it is in the case of uncompetitive inhibition. Option 1. The genes which remain confined to differential region of the Y chromosome they are called as Hollandric genes. Blastopore is opening of gastrocele. ARP, profilin and villin are all actin binding proteins. CDK1 can only be fully active when it is dephosphorylated by CDC25. Which one of the following statements about BT cotton cultivation in India is not true? Farmers cultivating BT cotton are benefited from higher return. Pesticide consumption of cotton is significantly reduced. India became an exporter of cotton. The cotton cultivation in India is free from bollworm menace. That is not true because even resistant bollworms have been detected, which are resistant to BT cotton. So you cannot say that it is free from bollworm menace. Then the latest version of golden rice contains the following transgene. So 2 genes, one each from maize and erwinia. Answer is option 3. Which of the following studies is not needed for the biosafety assessment of GM crops? Human and animal safety studies, yes. Gene flow analysis, yes. Through pollen grains, so that is needed. Soil microflora, natural enemies of the pest, yes. Micronutrient uptake by plants, so that is not needed for biosafety assessment of GM crops. Then, which one of the following steps is not true for production of artificial seed by desiccated system? Somatic embryos are first hardened to withstand desiccation. Hardened somatic embryos are encapsulated in a suitable coating material. Somatic embryos may be hardened by either coating or treating mature somatic embryos with a suitable polymer, followed by drying during their maturation phase. Somatic embryos can be planted directly in the field. So the answer is option 4, that is somatic embryos can be planted directly in the field. That is not true for the production of artificial seeds by desiccated system. You cannot plant somatic embryos directly in the field. Then absorption of which of the following nutrients in the human intestine is interfered by phytate present in the seeds? It is iron. Phytic acid interfere with the absorption of iron.